Hello everyone, welcome back to my channel Suggested Tutorials. Today in this video session we are going to discuss about prokaryotes. Already in the earlier session we have discussed that cells are divided into prokaryotes as well as eukaryotes on the basis of presence or absence of the nucleus. And now we are going to study the complete structure and details about the prokaryotes. Prokaryotes. What is the meaning of the word prokaryotes? The word prokaryotes is divided into two terms. Pro and karyotes. Pro means before and karyon means nucleus. In the prokaryotic cell, the nucleus is not completely developed. Not well-defined nucleus is observed in the prokaryotic cell and it is also not surrounded by nuclear envelope. So, well-defined nucleus is absent in the prokaryotes. Nucleus is absent. So, the nucleus which is present in the prokaryotic cell is only partially developed and it is not fully developed and it is also not surrounded by nuclear envelope. Nuclear envelope is absent. And also the cell organelles. What are the cell organelles? Mitochondria, Golgi apparatus, endoplasmic reticulum, lysosomes. These all are called as cell organelles. These complete cell organelles are absent in the prokaryotes. Cell organelles are absent. What are the cell organelles? Mitochondria, Golgi apparatus, endoplasmic reticulum, lysosomes, etc. All these cell organelles are completely absent in the prokaryotic cell. Whereas in the eukaryotic cell, these cell organelles are present. And one main point in the prokaryotes is the one and only cell organelle which is present in the prokaryotes are ribosomes. The ribosomes are protein-like structures, small vesicle-like structures. These structures are present in the prokaryotes and also present in the eukaryotes. This is only one and only cell organelle which is present in both eukaryotes as well as the prokaryotes. And next, the most important feature is the organisms which are involved in the prokaryotes are exclusively unicellular organisms only. There is no chance for presence of multicellular organisms. The organisms which comes under the category of the prokaryotes are exclusively unicellular. But whereas in the eukaryotes, both unicellular and multicellular organisms takes place. So what are the unicellular organisms which comes under the prokaryotes? Blue-green algae, bacteria, PPLO and also mycoplasma. So these are some of the organisms which are exclusively unicellular organisms. BGA means blue-green algae, PPLO means pleuropneumono-like organisms, bacteria and also mycoplasma. And coming to the multiplication or duplication process, the duplication process in the prokaryotes is very faster when compared to that of the eukaryotes. So duplication or multiplication process is very faster in prokaryotes. So, if a prokaryotic cell wants to divide or multiply, it takes a very short period of time for the process of multiplication and leads to the formation of the daughter cells. But whereas the eukaryotic cell requires much more time for the process to development of the younger ones and also for the process of multiplication. So, the duplication or multiplication phase requires very short period of time. And also, coming to the shape of the prokaryotic cell. Is there any particular fixed shape of the prokaryotic cell? No. There are very different types of shapes in the prokaryotes. So let us discuss about the shape of the prokaryotic cell. And now let us discuss about the shape or different shapes which are present in the prokaryotic cell. Coming to the shape of the prokaryotic cell, there is no particular fixed shape of the prokaryotic cell. There may be different types of shapes. At first, the prokaryotic cell may be round or spherical shaped. If the prokaryotic cell is round or spherical shaped, it is denoted as coccus. What is the meaning of the coccus? Round or spherical. If the prokaryotic cell is round or spherical shaped, then it is called as, mentioned as coccus. Or else, some of the prokaryotic cell may be spiral shaped or spring-like structures. The spiral shaped or spring-like structures are called as spirillum. And also, some of the prokaryotes may be in the form of rod shaped. These type of rod shaped prokaryotic cell are called as bacillus. So, if the prokaryotic cell is in rod-like structure, then it is mentioned as bacillus type. And some of the bacteria, some of the prokaryotes looks like comma shaped. Comma. So, if the prokaryotes are in this comma shaped structures, these are mentioned as vibrio. So, these are the most commonly observed different shapes of the prokaryotes. Some of the prokaryotes may be round. If the prokaryote cell is round or spherical like structures, it is called as coccus. Or if the prokaryotic cell is in a spiral or a spring-like structures, it is mentioned as spirillum. And if the prokaryotic cell is rod-like structures, then it is called as bacillus. Or if the shape of the prokaryotic cell is comma shaped, it is called as vibrio. So these are the different shapes which are included in the prokaryotes. And now we are going to study the complete structure of the prokaryotic cell by considering the bacterial cell. So this is the complete bacterial cell, and the outermost layer which is surrounding the complete bacterial cell is the glycocalyx. So the outermost layer of the bacterial cell is the glycocalyx, and the glycocalyx is followed by the layer called cell wall. And the cell wall, the innermost layer, the layer which is followed by the cell wall is the plasma membrane. Plasma membrane, or it is also called as cell membrane. So, the outermost layer is the glycocalyx, and the glycocalyx is followed by the layer called cell wall. It is the second layer, and glycocalyx is the outermost layer. Cell wall is the middle layer, and the cell wall middle layer is followed by the another layer called plasma membrane, which is the innermost layer. And the glycocalyx, cell wall and plasma membrane, these three combined and called as cell envelope. Cell envelope. The cell envelope is the three-layered structure. The outermost is the glycocalyx. The middle layer is the cell wall. And the innermost layer is the plasma membrane. The three layers combined and it is called as cell envelope. What is the function of the cell envelope? The cell envelope function is to protect the bacteria from the any damage or destruction. So this is about the complete layers of the bacteria. And also from the cell wall layer arises a tail-like structure or a filament-like structure. This tail-like structure is called as filament. Again I am repeating, from this layer of the cell wall, which is the middle layer, it arises a thread-like structure or a tail-like structure. The tail-like structure which arises from the cell wall is called as filament. What is the purpose of the filament? How it is used? The filament is used for the purpose of movement. The filament is used for the purpose of the movement. If the bacterial cell wants to move from one place to another place, then the filament helps for the process of the movement. And also along with the thread-like structure filament, the surface of the bacterial cell is completely filled with thin finger-like projections. These finger-like projections which are present on complete surface of the bacteria are called as fimbriae. Fimbriae. So, the tiny thin finger-like projections which arises on the surface of the bacteria are called as fimbriae. What is the function of the fimbriae? The filament is used for the process of movement. If the bacteria wants to move from one place to another place, filament is used. Then what is the purpose of the fimbriae? The fimbriae helps for the purpose of attachment to the rocks. If the bacteria want to attach to some of the rocks or any other substratum, the fimbriae is used. So, for the process of attachment to the substratum or rocks. If the bacteria wants to stick some of the rocks very sharply or very hardly, these fimbriae, finger-like projections help for the process of the attachment. And also, inside the plasma membrane, this is the plasma membrane. Inside the plasma membrane, a fluid-like substance is present. What is this fluid-like substance which fills the complete bacterial cell? Cytoplasm. So, the cytoplasm is the fluid-like substance which completely occupies the bacterial cell. What is the purpose of the cytoplasm? The cytoplasm undergoes different reactions. Chemical conversions also takes place in the cytoplasm, and also cell division also happens in the cytoplasm. So, what is the function of the cytoplasm? Chemical reactions takes place in the cytoplasm, and also cell division also takes place in the cytoplasm. And also the cytoplasm contains a circular ring-like structure. What are those circular ring-like structures? These are called as circular DNA. As we all know that DNA will be like thread-like structures in humans. But in the bacteria, the DNA, mostly important, note this point, in the prokaryotes, the DNA is in circular shape. So, the type of DNA which is present in the prokaryotes or in the bacterial cell is circular DNA. And also, along with this circular DNA, some of the circular bodies are present which are densely packed. Circular structures are present inside the cytoplasm which are freely floating. What are these circular bodies are called as? These circular bodies are called as inclusion bodies. So, these circular densely packed which are freely floating in the cytoplasm are called as inclusion bodies. What are inclusion bodies? The inclusion bodies are nothing but reserved food materials. We are taking food and we are storing in the form of fat, proteins, carbohydrates, etc. Similar in that way, the bacteria also stores some of the food which are called as reserved food materials. So, inclusion bodies are nothing but reserved food materials. Reserved food materials. So, along with these inclusion bodies, the surface of the The surface of the bacteria contains small protein-like structures. What are those small protein-like structures which are present on the surface of the bacteria? Ribosomes. Ribosomes. The ribosomes are the circular disc-shaped round protein-like structures which are observed on the surface of the complete bacterial cell. So, again I am repeating, the bacterial cell contains a fluid-like matrix called as cytoplasm. Inside the cytoplasm, circular ring-like structures are observed. Those circular ring-like structures are called as circular DNA. Along with the circular DNA, densely packed round, which are freely floating in the cytoplasm, round structures are observed. Those round structures are called as inclusion bodies. What are the inclusion bodies? The reserve food materials which are stored by the bacteria are called as inclusion bodies. Along with the inclusion bodies, the bacterium surface is completely filled with small protein-like structures. Those protein-like structures which are observed on the surface of the bacteria are called as ribosomes are present. And along with this, thread-like structures are also observed. Those thread-like structures are called as normal DNA. But along with the normal DNA, circular DNA is also observed in the bacterial cell. So, this is about the complete structure of the prokaryotic cell. And also, the plasma membrane contains some of the gaps. The plasma membrane of the bacterial cell contains some of the gaps. These gaps are filled with vesicle-like structures. So, inside the bacterial cell, the plasma membrane is broken down and it is some pore-like structures are arranged on the surface of the plasma membrane. What are those pore-like structures or vesicle-like structures which are present on the plasma membrane? These structures are called as mesosomes. So, mesosomes are the outcomes of the plasma membrane which are vesicle in shape or sometimes it may be like a lamella shaped also. So, the outgrowths of the plasma membranes are called as mesosomes. What are the function of the mesosomes? The mesosomes are helpful for the process of respiration or any energy conversion is also required, takes place in the mesosomes. And also the mesosomes are also used for the formation of the cell wall. See, this is the plasma membrane and the outgrowths are present on the surface of the plasma membrane. These are called as mesosomes and these mesosomes are helpful for the formation of the middle layer called cell wall, and it is also used for the process of the respiration. Respiration also takes place in the mesosomes. So this is about the complete structure of the prokaryotes. And now we are going to discuss about the ribosomes in the prokaryotic cell. So, what are the ribosomes? Where those ribosomes are present? The ribosomes are a site for protein synthesis. Means the proteins are formed or created here in the ribosomes. And also, these ribosomes are present on the surface of the plasma membrane. This was the glycocalyx, this was the cell wall. And the innermost layer is the plasma membrane. So, the innermost plasma membrane surface is occupied with the small vesicle-like structures. These small vesicle-like structures are called as ribosomes. So, the first main point is the ribosomes are site for protein synthesis. Here, the protein formation happens in the ribosomes. The proteins which are required by the bacterial cell is formed at the ribosomes. And also, the ribosomes which are present in the prokaryotes are exclusively 70S type. In the eukaryotes, 80S type of ribosomes are observed, but whereas prokaryotes, 70S type of ribosomes are observed. And also, where these ribosomes are present? The ribosomes are present on the surface of the plasma membrane. So, plasma membrane surface is the site for the ribosomes. So, where the ribosomes are present? The ribosomes are present on the surface of the plasma membrane. And also, the ribosomes which are present in the prokaryotes gets attached to the mRNA molecule. For example, this was the mRNA molecule. The ribosomes gets attached to the mRNA molecule and leads to the formation of chain-like structure. The ribosomes which gets attached to the mRNA and leads to the formation of chain-like structure are called as polysome. Poly means many. Here many ribosomes are attached to the mRNA molecule. So it is named as polysome. Polysome, or it is also called as polyribosome. I am repeating again. The ribosomes which are attached to the surface of the plasma membrane gets attached to the mRNA. So the ribosomes which are present on the surface of the plasma membrane gets attached to the mRNA molecule, and this mRNA molecule gets attached to the ribosomes, and here the chain-like structure is formed. This chain-like structure called as polysome or polyribosome. What is the function of the polysome or polyribosomes? So the ribosomes which gets attached to the mRNA molecules helps in the formation of proteins. So the ribosomes which helps in the formation of the mRNA which gets attached to the mRNA molecule are useful for the formation of proteins. So this was the complete details about the ribosome. And now we are going to discuss about the complete function of the cell envelope. Cell envelope. What is the cell envelope? We have already discussed that cell envelope is a three-layered structure. Again I am drawing the bacterial cell. From the cell wall arises a thread-like structure called as filament, which is useful for the purpose of movement. The outermost layer is the glycocalyx. The middle layer is the cell wall, and the innermost layer is the plasma membrane. At first, we shall discuss about the outermost layer, glycocalyx. What is the function of the glycocalyx? At first, the glycocalyx layer, which is the outermost layer of the bacterial cell, is prepared with a material called peptidoglycan. So, the glycocalyx is prepared with a material called peptidoglycan. And what is the function? Coming to the function of the glycocalyx helps us to divide the bacteria into two types. What are those two types? Gram-positive bacteria, gram-negative bacteria. So, on the basis of composition of the peptidoglycan, the bacteria is divided into two types, gram-positive bacteria and gram-negative bacteria. At first, what is gram-positive bacteria? If the glycocalyx layer undergoes staining process, then it is called as gram-positive bacteria, which means this is the outermost glycocalyx layer. If the glycocalyx layer contains thick layer of peptidoglycan, the presence of more thick layer of peptidoglycan leads to the gram-positive bacteria undergoes staining process. So the bacteria which contains thick layer of peptidoglycan undergoes staining process. If the bacteria undergoes staining process, what happens? Then it is called as gram-positive bacteria. If the bacteria contains thin layer of peptidoglycan, then it does not undergo staining process. If the bacteria does not undergo staining process, it is called as gram-negative bacteria. Don't be confused. I am repeating again. The outermost layer is the glycocalyx. The glycocalyx is prepared with the material called peptidoglycan. And if the peptidoglycan is very thick, then the bacteria undergoes staining process. If the bacteria undergoes staining process, it is called as gram-positive bacteria. If the peptidoglycan is very thin or slime layer, then it does not undergo staining process. If the bacteria does not undergo staining process, it is called as gram-negative bacteria. So, this is the function of the glycocalyx. On the basis of a composition of the peptidoglycan, the bacteria is divided into gram-positive as well as gram-negative bacteria. If the peptidoglycan is very much thicker, it undergoes staining process. If the peptidoglycan is very thin, it does not undergo staining process and it is called as gram-negative bacteria. This is the function of the layer glycocalyx. Coming to the second layer, middle layer, cell wall. What is the function of the cell wall? The cell wall helps and gives complete protection to the bacterial cell. If any damage or destruction occurs to the cell wall helps from the destruction or damage. So, what is the function of the cell wall? Protection. Protection to the bacterial cell is given by cell wall. So, any destruction or damage occurs to the bacterial cell wall helps. And coming to the plasma membrane. What is the function of the plasma membrane? The plasma membrane holds cytoplasm. We have already discussed that inside the plasma membrane, a fluid-like substance is present which is called as cytoplasm. And on the surfaces of the plasma membrane, we have already discussed that vesicle-like structures arises. What are those vesicle-like structures? Mesosomes. The plasma membrane contains vesicle-like structures which are called as mesosomes. These mesosomes helps in the process of respiration and also lead to the formation of the cell wall. These are the complete function of the cell envelope. So, this is about the complete details of the prokaryotes. Today we have discussed about the basic points of the prokaryotes and also we have discussed the structure of the prokaryotes, shape of the prokaryotes and also we have dealt with the complete function of the cell envelope. I hope you have a clear idea about the prokaryotes through this video. If you like this video, hit a like button, share to your friends and subscribe to Satyasree Tutorials. Thank you.